Hi Miriam, thanks so much for joining me for this next instalment of Charting the Best course, an interview series with the Farmer Navigator. First of all, could you please introduce yourself for our viewers and provide us with a little bit of background as to your industry experience and your current role?
Thank you very much. Delighted to be on the call this morning, Felicity. So, I'm Miriam Monge. I'm Head of Industry Advocacy Strategy at Sartorius. I've been with the company for over 25 years. I started off with STEDIM in the pioneering days of single-use technologies. And I've had the pleasure, I would say, of seeing and participating in the evolution of this industry over many years. Of course, in the early days, single-use technologies were not taken terribly seriously as an alternative to traditional manufacturing technologies, stainless steel, and so on. And of course, over the years, also through the different value propositions we've been able to develop, demonstrating the value of these technologies, we've seen a complete transition of the industry in this direction. So yeah, excited to be on the call today and looking forward to our discussion.
Thank you. So we're seeing that the biopharma landscape is transitioning away from those uniform blockbusters towards highly targeted therapies, complex bioconjugates, and those smaller kind of patient populations. From an industry perspective, how has this trend, this shift, altered the baseline math when planning multi-year capacity? Is the industry moving away from those dedicated lines, perhaps?
Yeah, absolutely. I think clearly what we've seen is that the blockbuster model that we saw for so many years, where you had companies manufacturing tons of proteins, so monoclonal antibodies, that blockbuster model is really dying, and we're seeing a strong shift away from that. So if you look at the molecule pipeline today, only 20% or less of the molecules in the pipeline require more than a ton per year, and actually 50% require less than 100 kgs per year. So of course that means that you're moving to the requirement for much smaller scale facilities. Linked with that, of course, we've seen increased titers, and also some of the complex molecules, the bispecifics, trispecifics, they don't work well in typical fed batch mode. To up the titers, you need to have intensified manufacturing technologies. So again, this means overall you require facilities that are able to manufacture multi-modalities, but considerably smaller quantities. In addition to that, we're seeing a patent cliff like we've never seen before. So over the next 10 years, we've got 50 blockbuster drugs that are coming off patent. And of course, to give you an example, if you take AbbVie's Humira, that represents 600 kgs per year, and they're now competing with 30 biosimilar manufacturers for those 600 kgs per year. So effectively, you see a strong shift towards smaller scale, highly flexible facilities in terms of the capacity landscape. So still about 20% will have those large scale facilities for the remaining blockbuster drugs, of which of course there are far less.
Yeah, absolutely. And looking at those modern state-of-the-art facilities, these require immense capital expenditure. What are the primary operational challenges in ensuring that a facility can pivot between complex modalities without facing extensive, downtime or cleaning validation bottlenecks?
Well, that is indeed the reason why we've seen the industry transition so much towards single-use technologies, because single-use technologies inherently bring that flexibility. You can rapidly reconfigure single-use technology platforms in a way you cannot with stainless steel facilities. One of the big issues with stainless steel facilities where often you've got a 3 to 5 year build time, and sometimes there's only about 10% of the molecules in the pipeline that actually make it to market. So sometimes the molecules for which those stainless steel facilities have been designed, didn't make it to market, and then they ended up having to be used to manufacture something else, which wasn't necessarily the perfect size or scale. So you often ended up with highly inefficient facilities. Whereas with single-use technologies, you can delay your investment decisions until considerably later, because the build time for a single-use technology facility is much shorter. Why is that? That's because you can effectively decouple the building of the facility. You could also use a modular facility, which is even quicker, and you have the design of a single-use process platform in parallel. So typically, you should be able to get a single-use technology platform up and running in 18 months, or in some cases even less. If you take COVID, for example, during COVID, we had to rapidly configure manufacturing lines for manufacturing the COVID vaccines. We had to do that in a matter of months. And if it had to wait for stainless steel facilities to accommodate that, it would have taken considerably longer. So that's the real value of a single-use technology platforms.
Retrofitting, is another approach that people take to try and keep those costs down. So rather than building, a greenfield site. But from your experiences, are there structural or facility level limitations where retrofitting can become more restrictive than just starting from scratch, do you think?
Yes, of course. So I mean, obviously, the disadvantage of retrofitting is that you can't benefit as much from the latest generation technologies. You may just be able to implement one or two process steps, so you end up with sort of a hybrid facility. Of course, again, when you're implementing from scratch new next generation technologies like process intensification, you can look at how you can downsize your water systems. You can considerably reduce the footprint of your process in the clean room, which means that you can downsize your HVAC systems, which of course are the biggest energy consumer in your facility. So there are, of course, multiple benefits of moving to a completely new manufacturing facility. It all depends on your timelines. If as I mentioned earlier, you can potentially implement in a new single-use based facility in less than 18 months. Even faster, potentially, if you're using existing modules, these modular facility concepts like those from GCON, for example. So it's really a trade-off, and the question is time. Then, if you've only got 6 months, and of course you're just going to implement those a few process step improvements, but with the risk of having a less efficient facility, because you'll probably end up with too large size water systems, HVAC systems, and so on.
And how then should executive teams balance the immediate pressure of clinical timelines with long-term commercial compliance when deciding on whether to modify their existing established site rather than breaking ground on a new facility?
Yeah, this is a question actually that was tackled by Dr. Rhonda Duffy, Chief VP and Chief Executive, Chief of Operations at Biocon during our PI forum in India just over a week ago, and she was talking a lot about the balancing, the science, the industrialization, and business imperatives. And of course, in the end of the day, the ultimate thing is speed. I mean, she's focused on biosimilars. And of course, for a biosimilar, those who get to market first get the lion's share of the cake, right? And so the speed is everything. So from her perspective, ideally, of course, you need to design in these next generation technologies already in process development, so that you can then rapidly implement and go to market with that biosimilar very fast and in the most cost-effective way. She explained, of course, that if you start retrofitting or even trying to go to a completely new manufacturing technology, inevitably you're going to run into hurdles, because you're going to, if that product's already in filing, you're going to then need to redo stability studies. You're likely to lose about 18 months, and then you may not be first to market or even second or third, and then you're only going to get the breadcrumbs, so to speak, at the end of the race. So that's the difficulty, and she talked a lot about the commercial challenges and the need for scientists and manufacturing technologies to understand the need to balance the science with the commercial imperatives.
Yeah.
So that business case is extremely important.
Yeah. A common misstep within industry is equating sheer tank capacity or square footage with actual technological capability. So, what are the risks a development program faces when an organization expands the physical footprint without matching investment in analytical depth and process development infrastructure?
Yeah, I think there was considerable risks. I mean, if you think about tech transferring into a new manufacturing facility, it often requires multiple process changes, which introduce risk to process performance and product quality. You can have many differences, like differences in seed train design, bioreactor scale up, chromatography setup. So really, those robust analytical capabilities are critical to be able to monitor the impact of those changes, and also those process development capabilities to be able to build and use downscale models, characterize a large scale process design. So what we're seeing more and more is we see increasing use of high throughput systems like the AMBER 250, as they provide scalable downscale models and enhanced PAT capabilities, process analytical technologies. Yes, so really you need to be able to reduce development and tech transfer risk.
And during the early stages, we see programs stall, as they scale because processes were optimized for that immediate speed rather than the long-term industrial manufacturing. What should companies avoid doing when transitioning a complex biologic or bioconjugate from bench scale discovery into that cGMP environment?
I think, well, clearly the scientists need to really start with the end in mind and think about the need to develop and design scalable platforms from the outset. We do see this, right? The worst that can happen is that technologies are designed to research scale and have only been customized specifically for that, and then they're not able to be scaled up and implemented into CDMOs, for example. So the examples really start at the choice of expression systems. So you need to think, for example, about the cell line history. Has it been well described? Has it already been used in approved products? Can it be cultured in suspension in large scale tank bioreactors? Is it suitable for process intensification? These are all the sorts of things that the scientists need to think about in the design of their process so that it's suitable to scale. Also things like media choice is critical. Is it animal component free? Does it not contain highly expensive or non-GMP available components? And and of course, all the manufacturing technologies, the bioreactor types, the cell retention technologies, the downstream processing technologies, they all need to be fully scalable. So that's the scientists need to think about the full industrialization of capabilities.
Brilliant. Thanks so much, Miriam. It's been absolutely wonderful talking to you.
Thank you. It's been my pleasure. Really enjoyed it. Thank you for this, too. Thank you.